Hello, people. Sorry I'm a little late. I had some important Facebook to catch up on there. Got sucked in. You know how that happens. Okay, quick thumbs up, thumbs down here. How many people do Facebook besides, is it just for old people? How many of you people, you Facebooks? Anybody else? Come on, are you there or not? Rocky does Facebook. Vanessa does not. Who else? Pepe Le Pew gives me a no. Angelique does it. Yay, Angelique. And the rest of you are not here. You're just leaving a hand. Rondo. Another yes from Audra. Esha Gupta? No. Okay, just curious. I'm wondering if this is an old guy thing or if real people still do it. Sandy Robinson says no. Michelle Simmons is in. Okay, so quiz today. It's open now. It's very, very short, so it's only open until 10 after. You already lost a minute. 10 after. So go do that quiz in Moodle, and we'll get back at 10 after. Do more Facebook. Do some more. Everybody's doing a quiz. Go do the quiz. It's in Moodle.
All right.
Kwame, there's a quiz. Go do the quiz in Moodle. And we're back. Okay, people, I hope you found that quiz very, very difficult, because that would mean you didn't study and you get what you deserve. All right, let's take a look at the quiz. If I can find it. Un momento, por favore. Per favore. That's Italian. Por favor is Spanish. Per favore is Italian. Joe Mama is New Jersey. Where the hell is this quiz? That's Southern. Okay. Quiz. Okay, these are adhesion molecules and adherens junctions. Well, that's not hard. That's your cadherin. This is the extracellular attachment for focal adhesions. Fibronectin. These are adhesion molecules for focal adhesions. That would be your integrin. This describes the nature of adhesion molecules in adherens junctions. Ooh, homo, they're homos. This describes the adhesion molecules in desmosomes. They are heteros. Okay, makes sense? I hope so. Oh, shoot. I'm sorry, I did it again. I didn't share the right screen. Okay, adhesion molecules and adherens junctions, those are cadherins. Extracellular attachment for focal adhesions is your fibronectin, which is extracellular matrix. These are adhesion molecules in focal adhesions, that's your integrins. This describes the nature of adhesion molecules in adherens junctions. They are homophilic. This describes the nature of adhesion molecules in desmosomes. They are heterophilic. So homophilic meaning it's the same cadherin from one cell and the other cell meeting each other, homo, same. And heterophilic is when you've got one cadherin interacting with a different cadherin, heterophilic. Okay, second question. Cells in the presemitic mesoderm pass the differentiation wavefront and become somites. That would be MET. Primitive streak cells changing and entering through the node. Oh, that would be ENT. Right? Epithermal. No. Let's see. Yes. Epithelial to mesenchymal. Yes. Primitive streak cells are going from epithelial into mesenchymal, and they're going through. Okay. The only cell-cell connections, these would have to be your gap junctions. The main cell-cell connections would be adherens junctions. Bum, 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 bum. I'm getting so burned out, I'm getting punchy here. Okay, any questions about that quiz? I've never had questions. Don't question me, I'm in charge. Actually, I'm in charge. You should ask me questions. Whatever. So let's do some learning as long as we're here. Let's move on and talk about something else. Just got a chat from someone. Chatster. How are you deciding how many bonus points we get? Are you going to add the quiz scores and for every 10 points give us one? Yes, that is correct. Oh, that was a direct message to me. So yes, how are we doing these things? Add up all your quiz points, divide by 10, and that's how many bonus points you'll get. And I will let Excel do the rounding, so don't ask me about rounding errors. Not responsible for rounding errors. Okay, everybody ready? Or who is Pepe Le Pew, for God's sake? Pepe Le Pew. Pepe Le Pew was funny in the 60s. Today, he's a sexual harassment person. What was I thinking about Pepe Le Pew? There was a real funny Pepe Le Pew thing. I don't know. Anyway. Okay, morphogenesis. So I talked about this last time, this experiment, but I'm bringing it up again today because it really kind of helps to explain how cells change during compaction, how they change their surfaces. So here we're taking a chunk of mesodermal cells and a chunk of ectodermal cells. We're going to hit them with something to diffuse them into individual cells instead of tissues. So probably, I'm guessing collagenase, because that will break down the collagen between cells, and that has a tendency to make them let go. So now we've got just individual cells. We put them together into one tissue culture dish, and we let them go. And what do? They rearrange themselves. So mesoderm is touching mesoderm and ectoderm is touching ectoderm. And here we've got the ectoderm, what's it say? On the ectoderm on the outside, endoderm on the inside. Really? This doesn't make sense to me because up here, ectoderm is blue, mesoderm is yellow, and here yellow's on the outside, blue's on the inside. So that could be wrong. But either the figure is wrong and I didn't make the figure, or the interpretation is wrong. I'm not sure. But what I'm trying to get across here, though, is that, you know, the mesoderm has got mesodermal cadherins on them, and the ectoderm has got ectodermal cadherins on them. And when these adherens junctions are made, they will cause the cells, same cells, to stick together because of that homophilic nature of the adherens junctions. So, you know, so say you've got, you know, mesodermal cell bumps into another mesodermal cell, grab it, you know, and then an ectodermal cell comes in, bounces into it, bounces off because it doesn't stick. So, you know, with time, they all stick together. And they're going to maximize the number of cell-cell contacts, and that helps to sort them out. So that's kind of what we're seeing here with compaction. So here we've got an 8-cell embryo. This is our mouse embryo that we talked about earlier. And you can see you've got a lot of unattached surface here right after the 8-cell stage. So most of the surface is not touching anything. But then you start putting out these adherens junctions, and you can imagine, you know, if you got a tissue like this and you've got adherens junctions holding it here, and it, you know, there's always movement. It touches, oop, there's another adherens junction. Now it's stuck here. Movement, oop, there's another attachment. So as these cells bump into each other, they maximize those attachments. And so here you can see there's more attachment than there was here. And as you go along, it gets to be more and more and more. And the cells who kind of get ahead of the group, and the ones who have the most cell attachments, will get even more cell attachments. Them that has gets, right? Money goes to money. So the ones that start out with the most cell attachment will get more, and they'll get pushed to the inside, or kind of sorted to the inside by this factor of getting more and more attachments. And it'll reach an equilibrium where some are on the inside and they're totally attached everywhere. And the other ones, you know, they just don't have enough to attach to, but they attach to each other really tightly. So that apical surface gets minimized. So you've got one minimized apical surface in all the outside cells, and the inside cells have got attachments everywhere. And this is by the 16-cell stage. And so now you've got, and whenever you've got cell-cell contact like that, you know, the cadherins are touching cadherins, which are attached to the cytoskeleton, and the cytoskeleton feels that, and you get actin myosin contraction, and you get surface tension. So on that cell, the surface kind of contracts and gets attention to it, and that helps to sort. And also, that's going to affect our hippo signaling. So if you remember back from the last chapter, if you've got all your surfaces attached somewhere, hippo is attached, it goes into those adherens junctions, and it's inactive. If you've got an open apical surface, then hippo can become active because that protein AMAT will come off come off the apical, no, start over again. If you're attached everywhere, then that AMAT protein will come off the surface and go to the adherens junctions and release hippo, and you get hippo signaling. If and that breaks down YAP, so you don't make CDX2 anymore. If you've got an apical surface that's open, then a MOT goes up there, HIPPO goes to the adherence junctions, and then you don't break down YAP, and so you get more CDX2, right? YAP and TEAD get together and make CDX2. So anyway, this is just another way of looking at that HIPPO interaction, what starts it, and it has to do with those adherence junctions, and kind of them that has gets kind of a thing, in that the cells that have the most adherence junctions will maximize that until they sort themselves on the inside of that morula, and then they're attached everywhere around. And then on the outside, you minimize the apical surface by attaching to each other more and more. You can imagine like two round balls come together, they touch at that tangential point, right? But if the tangential point then pulls itself in more, you end up with a flat surface between those two balls. Now imagine 8 balls and they're all doing that. That's what compaction is. Okay, I hope that made sense. So that is that. The next thing then is Xenopus gastrulation. Boy, we keep talking about this, don't we? I'm sure if you're in cell biology, you're sick to death of Xenopus gastrulation at this point. I love it. I can't get enough of it. Okay, Xenopus gastrulation. So my chemistry teacher in high school told me that teaching science is teaching a series of lies. First lie is the easiest one to understand, but then you go, well, it's kind of true, but it's not quite true. Here's more detail. And that wasn't quite true either, because there's even more detail and more detail. So anyway, when I was talking about gastrulation at first, I was like, these cells crawl and they go up across. Well, there's a lot more than that. It's more than just the cells crawling across. We also have, like we talked about with some of the other models, convergence and extension going on. And this has to do with cell-cell contacts, and that causes our morphogenesis here. So let's take a look at that. What's next? So, both in terms of the gastrulation and the epiboly, we're going to have some changes of cell-cell contacts that are going to change the shape of that tissue, which is going to cause these motions to happen. So first one I'll talk about is the gastrulation, and that has to do with convergence and extension. So I like the Play-Doh model of convergence and extension. So imagine you've got like a cylinder of Play-Doh, you put it on the table, you roll it back and forth. I'm sure you've all done this, maybe not for 10 or 15 years, but you've all done it. Roll that on the table, and the Play-Doh goes from a small cylinder to a long, thin cylinder, right? That's convergence and extension. Convergence is the Play-Doh is coming together and it's extending. So that's what convergence and extension is, more or less. And what does that have to do with gastrulation? Well, think about it this way. Oh, I'm sorry, go back up. So the mesoderm is a band around the whole thing, right? It's a band around the whole embryo. And that band is going to converge on that blastopore and squeeze through the blastopore. So it's almost like this bad boy over here. I'm sure I don't know about all of you, but some of you probably had this. I had one, where you extrude your Play-Doh through this little hole. So you put the Play-Doh there and you crank down on the handle and it comes out through that little hole and it picks up a new shape as it goes through that hole. Well, that's what these cells are doing. They're coming in from the side and going through that blastopore hole. So they're going from this wide band of material into a narrow tube of material. And that's your notochord and some other associated mesoderm through the middle there. Okay, so convergence and extension. And it sounds like that too. If you could hear it, it's really quiet, but if you could hear it, it has that kind of sound to it. Okay, so this is how the convergence happens, and it has to do with cell-cell contacts. So let's see, lamelliponem attempt to crawl on the surfaces of neighboring cells, pulling them in towards the directions of the arrow. So, okay, let me blow this up. So you've got kind of this band of tissue like this, and the cells of the band are all kind of coming together into a more narrow band. So they're doing that by the fact that the long axis of the cell is perpendicular to the direction that it's going to extend. And then as these cells come in, and it's doing this by cell crawling with the lamellipodium, as these cells come in, these cells get pushed up. And so all the cells are coming together and elongating out. And you can kind of see that in this transition from this thing to this thing, right? Same number of cells, but now it's more narrow and longer. So that's your convergence, cells coming together and extending out. See, convergence, extension. Cells are aligned perpendicular, they crawl on each other, pass between each other, and extends. Okay. And so here, this is the kind of thing that, the first time you see it, it's like, what? After a while, after you kind of learn how to imagine this in 3 dimensions, it makes a lot more sense. But here is our band of mesoderm. Let me blow this up again. Here's our band of mesoderm. This is the dorsal side, this is the ventral side. So over here at the dorsal side, you've got B here and A here. A is going to be at the dorsal pore lip, so that's going to be the leading edge of this gastrulation. As it comes in, okay, these cells aren't just coming around, they're coming up and towards that extension point, okay? So they're drawing in, going through the blastopore, and then towards the anterior side. Okay, convergence and extension. So just imagine this as like a piece of plastic that's warm and melted, and it's coming up and over, squeezing through the hole here at the dorsal lip and being forced up this way. And when it's going through like that, it's crawling across the ectoderm. So it's not just coming through the hole and going blah everywhere. It's going across the top of the ectoderm, the blastocele roof. Here's a picture of it. Here's your endoderm, your blastopore here. And these cells are all coming this way and then in and across the top there. And so here's another one. This I just picked up on this now, and it's a little bit annoying. In this figure, you've got B and A, right? And B goes forward while A stays here. So B ends up in the anterior end. Here they've done the opposite. So here's B and here's A. A is going to go through your blastopore lip. B is going to stay where it is. So B will end up posterior mesoderm and A will be anterior. So pulling back the ectoderm so you can see the rest of the mesoderm, the middle mesoderm here, and endoderm here. So the interesting thing about this is, see how C is over here, and at the opposite side is D, C and D, and these are part of that middle mesoderm, the somites. So the notochord is going to go through first. So switch these two. Say this is B. B is coming in through and is going to be pulling C and D with it. So when it totally extends out, C and D end up next to each other. They started out at polar opposites of the cell. They come in through like this, and now they're together. That's part of that through the hole and extending. And the A coming in this way and up across here. The blue is the ectoderm, so that A is crawling across the top of that ectoderm. Convergence, oops, that's not what I wanted to do. What is that anyway? Yeah, okay, that's a pibuli. So what's going to make this happen? And kind of the interesting thing about the letters are A, B, C, D is that, at the beginning, A and B are next to each other, but A moves in and B stays. So now at the end, after gastrulation, they're at opposite poles, versus C and D are at opposite sides and they end up together. So the whole thing kind of smears up that way. Now those involuting cells then are only going to go across that blastocele roof. And why is that? Again, that has to do with cell-cell connections as well. Between epithelial cells, between mesodermal cells, you've got adherens junctions. So as those cells intercalate and form a line, they're still attached to each other by those adherens junctions. So as the front ones crawl, they pull the back ones with them. And those mesodermal cells are also connected to the endodermal cells that are under them. Okay, so you've got like a double layer thing going up and through. And those endodermal cells are connected to the mesodermal cells. I don't know 100%, but I'm assuming it's by desmosomes, the different cadherens from two different cells grabbing each other. So that becomes a coherent sheet, bilaminar sheet, going up and through the ectoderm. Now, it's not attached to the ectoderm. If it was, it wouldn't move anywhere, right? If you had desmosomes with the ectoderm over here too, it wouldn't go anywhere, but it's not. Over here, the only connections between the mesoderm and the ectoderm are attachments to the extracellular matrix on the floor of that blastocele. So imagine, you've got that layer of tissue across the top. Where are we here? I wish I had a picture of a blastocele. Blastula. Okay, so here, this layer of tissue, well, let's look at it here because we still have a blastocele here. You've got a layer of tissue here, which is actually 3 cells thick. And then you've got this fluid-filled cavity, right? Underneath this, between the cells in the cavity, is a layer of extracellular matrix, fibronectin, collagen, things like that. So that's what these crawling cells are going to go across. They're going to use cell crawling with focal adhesions and crawl across that. So that bilaminar thing of tissue here, you've got ectoderm and mesoderm are attached to each other. They're grabbing the extracellular matrix on the ectoderm and pulling themselves across it by cell crawling, right? Focal adhesion grabs, cell pulls forward, focal adhesion grabs, cell pulls forward, and so on as it goes across the top of that embryo. And the fibronectin along that way is all polarized. So it's all pointed in one direction that gives the focal adhesions and the crawling of these mesodermal and endodermal cells a direction to go in. So they're not going to come in and go in all possible directions. They always go in that one direction along that directional fibronectin. And so it makes that posterior anterior axis. Okay, any questions about that? Comments? Does that make sense? Nobody's chatting me. Chat. Okay. So, all right, this is kind of hard, but I have to tell you about another lie I told you earlier on. And that was about epibole. So early in the blastula formation, this is true. I said it at the time, and it is true. Your endodermal cells divide slowly, and your ectodermal cells divide quickly. So the cells on top, the ectoderm, you get many small cells versus the endodermal cells are few and large. So you get a whole lot of ectodermal cells there. I think 3 layers basically. So when epibole happens, I think earlier I said epibole was the cells dividing and coming over, but actually they're not dividing. It's another type of convergence and extension, except they're not like extruding through the blastula. They're converging radially, so the 3 layers of cells are getting thinner, and as that thins out, it stretches out this way. So let's see that. Here's a really cool electron micrographic look at the ectoderm, and they've colored them different colors just so you can see them better. So this is in the blastula. You've got 1, 2, 3 layers of cells. So here's a kind of a diagrammatic way of looking at it. 3 layers of cells. Now, these cells will crawl in this direction, and these cells are crawling in this direction. So they're crawling across each other, and as they do, they're displacing the surface cells or expanding them out, I guess. I guess these are the ones that are intercalating, and so they end up as 1 layer, whereas they were 2 layers. And then these by cell-cell attachments are now surface tension pulling themselves apart. So it's more like this. It's more like a it's more like rolling out pie dough. Imagine you take your instead of rolling it flat, you take a bowl, upturned bowl, right? You put a blob of pie dough on top of it, and you squish it down over the bowl. So it's thinning out and wrapping over the bowl. And that's what's happening here with the Pibili. They're not dividing, as I said before. I apologize for lying, but you guys like to be lied to, I think. Some people just like to be lied to. So these cells are thinning out and stretching over, and that's what's going to make them go from the animal pole all the way down to the blastopore. Okay? Radial intercalation. So that means they're going out, radially with respect to the circle along the radius. Okay. Questions about that? Comments? Discussion? You people are so easy. All I do is sit here and talk my BS and you listen to it. Let me show you a video talking about this that I think is really good and it's worth watching. Gotta find it first. That's not it. Here it is. All right. This guy is like way smarter than me, if you can imagine. I don't know, maybe hard to imagine, but he does a really good job with this. So let's watch this.
My name is Richard Howland, and this is the second in the series of talks about Xenopus gas relation acid formation. In this short video, I hope to show that gas relation, which is often thought of as a horrendously complex process, is actually fairly simple. You just have to take it apart and look at the individual cell behaviors. And quite a few different cell behaviors. These all conspire to get the whole mechanism of gas relation. So let's first start with this schematic. Solve the first problem. So what we're going to discuss is all of the component movements that go into this complex process of gas relation. So we'll discuss, for example, the crawling of cells across the blastocyst and other individual motions that contribute to the process. Okay, so here's a schematic which shows 7 basic mechanisms of gas relation. I'm going to discuss 6. We'll take them in turn. So the first one is the epibole, that is the thinning and spreading of the blastocyst. So all of this blue tissue. Of course, the blue tissue has to spread and cover the whole embryo. And recently, there's been a lot of progress made in understanding how this may occur. Epibole consists of this initial gathering of cells. These are freeze fracture electron microgases from that blastocyst. You can see at the beginning of gas relation, you have about 3 cells thickness on the whole surface. During gas relation, there's a thinning process. You can see these green cells here interlacing with one another. That, of course, is going to lead to a spreading. So there's a spreading not only on the green part by virtue of this rearrangement of cells, the interrelation between each other, you also see that these red cells begin to be a little stretched, no longer quite so cuboid. It's subtle, but it makes a difference in the overall process. Below here, it's represented schematically, where you start with the 3 layers and then go through this formation, and then a much wider array. This is the work of Richard Howland. What they found is that there's a signal that's made in the red cells, actually complements it. That acts as more attractive. Green cells act against the red cells. So that's the major driver of this spreading process. on to a second signal now, vegetal rotation. So vegetal rotation is the process whereby all of this Yogi stuff, you remember, has to constrict, live inside. So what we're going to see is there'll be a spreading of this flora and plasticine, while at the same time, the outside layer will have to collapse down essentially to a point to get enveloped by the rest of the air. That occurs by a fountain-like movement of cells. It's also slightly asymmetric, such that this dorsal side, which is head tissue up here, is going to end up moved against the blastocyst roof. You can see this flap here. So unlike on the ventral side, the dorsal side has this cleft, the plasma brachialis, that letters in contact with the outside part of the area. So again, let's watch what happens here. You can see the spreading there of the blastocyst roof. It becomes quite a large surface area before it finally collapses. The collapse of the blastocele is thought to be mediated mostly because these cells here are very loosely attached, and the liquid moves from the blastocele into this other cavity, the arcantron, which is the primitive gut cavity. At the same time, you can see the cells down at the bottom initially cover a very large surface area, but they collapse down to a small point. Again, this is thought to arise as a result of the rearrangement of cells, so there's this sort of fountain spreading of cells. This has been studied recently by Rudy Winkelbauer's lab, and here he's taken a slice of that Yogi tissue and put it in culture, and you can see there's a fountain-like movement of cells. So these cell rearrangements are exerting force, and here you can see also the spreading of the blast to the floor, whereas the initial area was much broader. So that's motive again going this vegetal rotation. The underlying mechanisms are not understood so well, but at least the cellular behavior is pretty described. Let's move on to bottle cell formation. We saw that as in the initial pigment line, which we can see from the surface of the embryo. And actually, we see these after there's already been quite a lot of action inside the embryo. So these internal movements have already started to occur. And these bottle cells form slightly later. And then as I mentioned, plus actually more attention structure. So when other forces extend this red pizza, and all of the forces of the rather than going outside the embryo, here's a light micrograph of a stained section by Jen Lee in the lab. And what she's done here is to stain the cell with tubular protectin. And as is generally found with these constricted cells, like the bottle cells, the old-fashioned glass and blood bottles, you see this apical constriction in the cells above the organ. And actually, the whole structure is quite aggressive. While the apices constrict, so you have this sort of red line, and then you have this white constriction. The actin-myosin contraction is mediated that constricts these apices and leads to the beginning of an invagination movement on the outside. There's a movie that shows that from the outside. So this is an embryo that's been labeled with a membrane-targeted GFP. And so what we'll see as this plays through is that up here there's not much action. These cells are perhaps being stretched. Down here where the bottle cells are forming, you can see the constriction of the apical surfaces of these cells. And it's by no means completely uniform. There are some cells in between them that are not undergoing apical constriction, and there's sort of passive distraction that forces from the constriction. And ultimately, all of these cells will start to apically constrict. That will form the existing cells to move inside the embryo and help the process of evolution of the management. I've already mentioned that this cleft here, the cleft of Brachiae, and of course this cleft could have two fates. One is that the tissues can remain separate, or they could merge with one another. And it's a real active process of tissue separation of these cells are.
Okay, so that's really all I wanted to show you. But this is what he's talking about here is basically what this chapter is about. We're not talking about signal transduction and hormones and new genes getting turned on. We're talking about physical forces that are attaching cells and contracting and causing them to move and make these shape changes, morphogenesis. So that's what's going on here. So I remember what I was going to say about Pepe Le Pew. The funny thing about it was like every single episode was the same. It was like this black cat who somehow got a white stripe on it by accident, and suddenly Pepe wanted to meet this young lady. So the cat, because it's French, the cat would go, "Les mieux, les mieux," because that's the way cats talk in French. Okay, I guess it wasn't that funny. And we talked for like 2 minutes after class. Angelique. Okay, Angelique, let me send you a link. Okay, so give me a minute. I've got someone to talk to at 2 o'clock and someone at 2:30. So I'll send you a link. We'll talk at 2:15. Okay? Okay. All right, that's all we got. I hope to see you again on Friday. Later, dudes.